Okay, so we know that the DNA is too big to go out of the nucleus to actually tell the ribosomes what to do. So it needs to make a messenger. So the process of making this messenger is called transcription. So transcription just really means it is the process of DNA making an mRNA strand. So making a messenger. The messenger is called mRNA. So how does the DNA do that? So let's start again. We know that DNA is a double helix. Okay, now this is where the confusion starts, because the process of transcription looks very much like DNA replication, but I'm going to show you just now what the differences are. So the first thing that'll happen again is that we're going to have some unwinding, but the difference between replication and transcription is that we have unwinding of the DNA partially. We don't unwind the whole DNA strand, which means the DNA strand, when it unwinds in a little section, it looks like this. It's going to be windy, windy, windy. A section opens up, windy, windy, windy. Section opens up, and then wind, 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 wind. So only a little part of it opens up, not the entire DNA strand. Just a section, partially, it unwinds. What will happen when that unwinding happens, after that's happened, we're also going to have the unzipping thing. Unzipping. Blech, sorry for the ugliness. And again, it's partially. It's not the whole thing. Windy, windy, windy. Open legs, windy, windy. Windy, windy. Open up. So it kind of just a section. All right. And that opened up section is then going to look like, you know, exposed G's, C's, T's, A's, whatever, like we had in the previous with the DNA replication. So let me make that picture a little bit bigger. Windy, windy, windy. Open up. Windy, 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 windy. Wind, wind, wind. Open. Wind, wind. Oops. Wind, wind, wind. Okay. So this is what we have there. So imagine we had a G, T, A, C, G, G, A, T. Let's say we had this on the one side. We know this is a DNA strand, so on the other side, G would have bonded to a C, T to an A to a T, C to a G, C, C, T, A. Cool. So it looks very much like DNA replication, but except the difference is the whole thing didn't unwind or unzip, hey? There it's still a double helix. There it's still a double helix. Just a section unwound. Now, why just a section? The reason for that is whatever the protein is that we want to make, that information is contained in the DNA, somewhere in the DNA. So if we reverse back and we look at this DNA strand over here, perhaps that first section over there contains the information for eye color. And then further on, that section over there contains the information for hair color. So as the DNA strand, as we run along the DNA strand, we find various sections of the DNA strand contains information for various traits. These sections are called genes. So we say that the DNA strand contains genes. A gene is a section that codes for specific information or for specific proteins, which means whatever the protein is that we want to make through transcription, only that gene will open up. So this section, the section that has opened up, that section is actually called a gene. All right, so that's why you don't open up the whole DNA strand. Now, when this happens, all right, again, we now have two exposed bases. We have two exposed DNA strands. And remember, same as with DNA replication, DNA doesn't like to be single, all right? So something weird happens. Instead of like DNA replication, both sides attracting loose bases, what will happen is that one of the sides will start attracting bases, not both sides. Now, in a paper, it's sometimes the left side, sometimes it's the right side. There isn't a given rule of which side it's going to be. So let's use a purple pen, and we imagine we are now going to attract loose nitrogenous bases, all right, in purple, loose bases. But what we're going to do is we're going to build an mRNA strand. Now, something about mRNA is that it's an RNA molecule. Now, RNA is unlike DNA. It doesn't have the deoxyribose sugar. There's a D. It actually has a ribose sugar. It's a different kind of sugar. The mRNA strand is also just a single strand. It's not going to have two sides. It's going to just be one halfy. It's also going to be shorter, and then something very significant is that you will have uracil or U's, never T's. Which means if I use this strand on the left to build an mRNA strand, the G from the DNA strand is going to attract a loose C. The T is going to attract an A. The A is going to attract a U. Because we are building an mRNA strand, not another DNA strand, you won't have a T there. You'll have a U there. C will attract a G, G a C, C a G, A, U, and TA. All right. Which means now this purple strand that we are forming on the inside here, this purple strand is actually the mRNA strand. Okay. What will happen, and this was step 3. This, what I've drawn over here, is step 3, where you have loose bases building an mRNA strand. They're assembled this way. But always keep in mind, because it's an RNA, you don't have the U's, you'll have T instead. Okay. As soon as that strand has formed, it will detach. In other words, that is what happens at number 4, step 4. The mRNA detaches, which means what will happen is that the DNA strand that looks like this, windy, 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 open up, windy, windy, open up, is still going to look like this for a moment. But that purple strand as a whole, it detaches and it moves out. So you actually find it separate over here. It's a G, A, U, G, C, C, U, A. Okay. So it comes off. It's loose. It comes off. And since it's separate to the DNA strand, what can happen is that this DNA strand at step number 5 will just wind up again. DNA wind up again. Which means once the process is done, the DNA strand is actually unchanged. It's just going to wind up again and look the way it looked like when we started. There's no change in the DNA. It just made a little opening, made a purple little strand which came off, and then the DNA just closed again. All right. What will happen, however, with this mRNA strand is that it's going to move, all right, mRNA moves to the ribosome. Remember, it's the messenger. So it's going to go to the ribosome to actually now tell the ribosome what to do.